Um, well, my name is Chris Curry, and I am assistant professor at Iowa State University, and I focus on greenhouse and controlled environment crop production. And I'd like to start by saying a few thank yous. First, I'd like to thank our sponsor, Fine Americas, for sponsoring these webinars and helping us bring this information to you. I'd also like to thank Brian Whipker for all the organization he does for these e-GRO webinars and trying to bring together all of us faculty members to bring you good information on greenhouse production. So thanks for your efforts there, Brian. So containers in horticulture. We've had containers in horticulture for actually quite a long time. Originally, the original containers were primarily ceramic or terracotta, and these go back to Egyptians would have containers to grow plants in. Wood were also some of the first containers that we were using. But really, the thing that changed greenhouse horticulture containers the most was the introduction of the petroleum plastic plant containers. And there's a number of reasons why they're great. One, they're lightweight. Two, they're easy to transport. Three, they're flexible. Four, they hold in moisture, unlike say the porous ceramic terracotta or wood. So there are a lot of benefits to plastic containers, and there's a reason why they've been adopted so widely in horticulture. However, there is a downside to using plastic containers in horticulture. And here it is. I think everyone is starting to become more sustainably minded and ecologically conscious, and I think it's hard to look at the amount of plastics that we use in horticulture and greenhouse crop production specifically, and try to figure out some way to deal with this or some alternative solution to these plastic containers. Well, one of the things that has increased in popularity, and as we're looking for alternatives to plastic containers, are biocontainers. And here we have just a couple of different of the commercially available biocontainers. Moving from right to left, we've got a peat pot, a peat biocontainer. Then we've got a coconut coir container, followed by a paper fiber container, and then finally a rice hull pot. And so I think that these are things that you've seen, particularly peat pots, have been around for a while. The rice hull containers and the coconut coir and are some of the newer containers. And the fiber containers, you may use some for hanging baskets, but again, they're being produced in a 4-inch, 6-inch, and #1 trade gallon. So these are some of the materials. And when we say biocontainers, they're going to be containers that are made with biological material, biodegradable material. The idea for these containers is that they will break down in the waste stream. Now, I'm not going to say anything about planting these containers with the plants still in them in the ground. That's a whole nother discussion for another day. But again, the thought is that consumers can take these containers and dispose of them sustainably, whether it's in their home compost or incorporating them into their garden bed. And that's really one of the primary reasons why we're interested in these biocontainers. But there are more biocontainers than simply these more traditional materials. We also have bioplastic biocontainers, and these are going to be plastic in the sense that it's a bunch of small units linked up to make a large plastic unit where you've got the monomers become linked up together to become polymers, but these are going to be plastics that are made from biological materials such as polylactic acid or polyhydroxide alkanoate. There's not going to be a quiz later on any of those names, but these are some of the new materials that are coming out. One of the reasons that there's some interest in these containers is that you're able to mold them on injection molding machines, much like traditional plastic containers. Also, they look similar to traditional plastic containers. Some people don't necessarily want to have that overly earthy look that some biocontainers have, whereas these biocontainers can have a much more traditional look. They work on filling machines and other automation in the greenhouse. Now, anytime we have a new technology that's introduced into our greenhouse production or our production techniques, we have to adjust our plant culture, okay? And here is one of the problems with irrigating with biocontainers. Some of you maybe have seen this on some other biocontainers. Now, if you look at those containers, they're not real great. The reason being is because these containers are designed to break down, sometimes that can happen during production. So what's one of the adjustments that we make? Well, we start to grow drier. So here we have an example of a bioplastic container that was irrigated under standard conditions and one irrigated under drier growing conditions. And you can see that modifying our irrigation practice is going to help us adopt that biocontainer into production and still produce an acceptable looking container for our consumers. Well, this is where we come to plant growth regulators. We all know that plant growth regulators are widely used in greenhouse crop production. That's likely why you have registered for this seminar. You know how important PGRs are in suppressing excessive growth and allow us to produce a plant that is proportional to the container to make an aesthetically appealing unit to sell. So here we have some New Guinean patients, and you can see again, applying about 5 parts per million of fluoropyrimidol or top fluor is going to produce a New Guinean patient in that 1801 cell that is properly balanced. But when we're using plant growth regulators, again, there are going to be factors that affect the efficacy of PGRs. For example, cultivar. Now here, we don't have an example of an untreated plant on the right and a treated plant on the left. Rather, we have 2 separate cultivars. One on the right is going to be a standard Angelonia cultivar, whereas the cultivar on the left is going to be a compact or a dwarf Angelonia cultivar. So we know that we're going to have to adjust our plant growth regulators for differences in cultivars. We also know that our media, our substrate, can affect the efficacy of PGR applications, specifically drench applications. So as you can see here, on the left-hand side, we've got a pansy that was grown in standard peat and perlite media and was left untreated. Alternatively, when you look at the 2 pansies on the right-hand side, one grown in peat and perlite media and the other grown in a peat and bark media, and both treated with a 1/2 part per million paclobutrazole, you can see that that plant treated with peat and bark is not getting the same growth regulator effect as that plant grown in peat and perlite. The reason being is the bark in that substrate is tying up some of your active ingredient, and it's reducing the efficacy of your PGR. So we know that we need to adjust our drenches when we're growing with different types of substrates. So the question that we had working on this is, do PGR concentrations need to be adjusted for plants that are grown in biocontainers? Again, I showed you that example of how we might need to modify irrigation to keep our containers looking good for retail. And I also showed you some examples of how we may need to adjust our PGR applications based on substrate or plant cultivar. But we wanted to look at the interaction of biocontainers and plant growth regulators. So the objectives of the research that I'm showing you here today is investigating how biocontainers affect the efficacy of these PGR treatments. Again, with the increasing popularity of biocontainers and the importance of PGRs in production, we wanted to try and see how you might need to refine your culture in order to successfully adopt these biocontainers into production. So some materials and methods. These are the 8 different biocontainers that we included in the study. And again, you see that same shot of the 4 more standard biocontainers: coconut coir, peat pot, paper fiber, uncoated, and I'll come back to that, and then that rice hull pot. Now on the bottom row, we also have a couple of those containers that are bioplastics. The PLA and lignin, so that's going to be 90% polylactic acid bioplastic, and then 10% of a lignin filler. And then our other container is PLA and soy protein, 60% PLA plastic biopolymer and 40% soy protein. And then the next container, you see, it's a paper fiber container. However, one thing that we did with this paper fiber container is that we dipped it in a biological-based polyurethane coating. And what we were doing there is to try and reduce the porousness of our container and increase the rigidity. And then finally, of course, we included our petroleum-based plastic container. That's our control, the industry standard. Next, we selected 2 different plant species. We selected Serena Angelonia and Purple Wave Petunia. We thought those would be 2 good model plants, both annual bedding plants. Like Dr. Whipker said, we're focusing on annuals in this presentation. The Angelonia has an upright habit, and we've got a Petunia which has that prostrate spreading habit. So what we did is we applied 2-ounce drenches of paclobutrazole or piccolo 10 days after transplanting our 288 cell seedlings into the containers. Plants were then treated with the solutions of 0, 1, 2.5, 5, or 20 parts per million. And again, those drenches were applied 10 days after transplanting our seedlings. Now, a little bit about the greenhouse conditions so you know how we were producing these plants. When we produced the Angelonia and we're running that experiment, we ran a 73-degree day and a 68-degree night. A little bit warmer for Angelonia. Alternatively, for the Petunia, we ran a 70-degree day and a 66-degree night. So we had a little bit cooler air temperature for growing the Petunia. For both of the plants, we targeted a 12 to 13 mole daily light integral. And that's the target daily light integral for an acceptable or good quality greenhouse floriculture crop, looking across a wide range of species. Now, since we were dealing with a growth regulator study and we were going to see some plants that were going to be smaller than others, we selected a fertilizing regime that would try to minimize differences in fertilizer applications between different plants. So what we did is all plants were fertilized once per week with 150 parts per million nitrogen. Okay, so all plants got thoroughly fertilized once a week with that solution. And then in between fertilizer treatments, we irrigated with clear water, but not to the point of leaching in between those fertilizer applications. So our goal was, again, to be providing the same total amount of fertilizer to each container. So those untreated controls would obviously require more irrigations than, say, those plants treated with 20 parts per million. But again, by thoroughly irrigating once per week, then using clear water in between irrigations without leaching, hopefully we had very minimal difference in the overall nutrition provided to those plants. So here we have a shot of our plants in the greenhouse, and this is when we were growing our Angelonia. One thing that we did after doing a preliminary experiment is we did have to block our experiment and put all of the plants that were in a certain container type together. So here you can see we've got the peat pots, or the paper fiber pots in the forefront, the coconut coir pots in the middle, and then on the far end are plastic pots. The reason we separated them is because there were some differences in irrigation, and that will come into play later in this presentation. So what did we say? We'll start out with Angelonia. We did see an effect of container type on the time to flower. Compared to our petroleum plastic control plants, or the plants that were grown in petroleum plastic, we had a delay of about 2 days for plants grown in the PLA and soy protein pots, as well as the paper pots that were coated with polyurethane. We also saw a 3-day flowering delay for plants grown in peat pot. However, while statistically significant, a 2 or 3 day delay in flowering is not something that we consider to be overly commercially significant. It would be if we were closer to 5, 6, or 7 days. But again, 2 or 3 days is not something we were too concerned about. One thing we did notice is that across container type, we did see a little bit later flowering as PGR concentration increased. And this is a phenomenon that is observed with certain species and active ingredients. Again, you see that plants treated with 0 to 2.5 parts per million, the Angelonia flowered in 34 or 35 days. That increased to 36, 37, and 38 days as we treated with 5, 10, or 20 part per million drenches, respectively. But again, here, our maximum delay in flowering was going to be 4 days. So we don't see a huge change in crop time in response to that PGR. But what we're most interested in is the size of our plants. So here we have our Angelonia. Now, if you take a minute and look at this, what we've done is we've arranged this graph, or this figure, so all of the columns are going to be our paclobutrazole concentrations, 0 through 20 parts per million. And then our rows are going to be those different biocontainers. And then on the bottom, we've got our petroleum plastic control. So let's just take a look at some of the results that we saw. So regardless of the biocontainer, I've just highlighted a few different ones here. As our plant growth retardant concentration increased, the Angelonia plants were smaller. And this is what we would expect in a dose response for plant growth regulators. We would expect increasing control as the concentration of PGR increased. So again, we expected that result. But we're more interested in the differences between containers. So let's take a look at Angelonia treated with 5 parts per million paclobutrazole. All of the plants that are included in these orange boxes were similarly sized when treated with 5 parts per million. Alternatively, when we look at those plants that were treated with 5 parts per million, and they were grown in either the coconut coir or the peat pot, they were smaller than the plants grown in other biocontainers and treated with that same concentration, 5 parts per million. So there we started to see where plants in those containers were smaller when treated with similar amounts of PGR. We saw that similar trend, say, when we go up to 20 parts per million. Again, all the plants in the orange box were very similar. However, those plants in the coconut coir and peat containers were smaller than the other plants that were treated with 20 parts per million. What's interesting, though, is when we look at the untreated control plants. Again, those plants in the orange box were similar. So we saw a lot of similarities for plants that were grown in many different biocontainers, but untreated. However, just like we saw in the previous examples, those plants in the coconut coir in the peat containers were smaller than the other plants when treated with no paclobutrazol, so just that straight deionized water, okay? Let's take a look at Petunia now and see what kind of results we had here. So again, we've got our different container types in our time to flower. And again, we saw a similar difference where we saw some variation in time to flower. Compared to those petroleum plastic containers, we had a 3-day flowering delay for Petunia grown in coconut coir or peat containers. Interestingly, plants grown in the PLA soy protein, the paper fiber, both coated and uncoated, in rice hull containers, actually flowered a little bit earlier than those plants grown in the petroleum plastic container. But again, although these are statistically significant differences, when we look at up to a 3-day flowering delay or 3 days earlier flowering compared to petroleum plastic, it's not an overly commercially significant impact. Similarly to Angelonia, we also saw that the increasing PGR concentration delayed the time that Petunia took to flower. Again, when we treated petunias with 10 or 20 parts per million of paclobutrazol, we saw a 2 or 3 day delay. While statistically significant, again, this is not overly commercially significant. But again, we want to know what about those plants and what do the plant sizes look like. So here again, we have that same figure. We've got our columns and paclobutrazol concentration, 0 to 20. And we've got our different containers with the petroleum plastic at the bottom. Again, as we look across these containers, as the concentration of paclobutrazol increased, plants got smaller. So again, the PGR was effective. But when we compare within a paclobutrazol concentration across containers, we saw similar variation. Those plants grown in PLA lignin or PLA soy protein, again, the paper fiber pots in the rice hull, were all similar in size to petroleum plants grown in petroleum plastic and treated with 5 parts per million. And we saw that same trend where those plants grown in the coconut coir and peat pot were smaller than those plants grown in the bioplastic, paper, rice hull, and petroleum plastic. And we saw that same trend, again, just for example, looking at 5 parts per million, and again, at 20 parts per million. But similar to the Angelonia, we want to look at those plants that were untreated. And we saw the same instance where the majority of containers were similar to petroleum plastic, except for those plants grown in coconut coir or peat. Those plants were smaller than the plants grown in petroleum plastic, even when untreated. So what does this all mean? Several biocontainers were very similar to plastic. Again, the plants grown in bioplastics, rice hulls, and paper fiber, including both the plain paper fiber and uncoated paper fiber, responded similarly to the plants grown in plastic containers when PGRs were applied. So the take-home message is, when you're using these biocontainers, there were no or very, very few differences across containers within these containers across PGR concentrations. So you don't need to adjust your PGR concentration for those containers. However, we had a different result with the coir pots and the peat pot. Both the plants, Angelonia and Petunia, grown in the peat pots as well as the coir containers and treated with paclobutrazol, were smaller than the other treated plants at the same concentration. However, keep in mind those untreated controls were also smaller than plants grown in the other containers and provided with no paclobutrazone. So we believe that these smaller plants are likely attributed to substrate moisture. One of the things that we did is, again, I mentioned earlier how we blocked our plants by the container type. That's because in our preliminary experiment, we saw that the plants grown in coir container and the plants grown in peat containers required more frequent irrigations. And it wasn't that the plants were using more water. We believe that it was due to a quicker dry down of the soil or of the substrate moisture. So what we're seeing there is, again, those plants are more porous and they're drying down more quickly. What we believe happened is that for those plants that were grown in the peat and coir containers, that porous nature actually imparted a slight moisture stress. We all know that when we grow drier, our plants don't grow as big. This is something that we see when we grow tomatoes dry in the spring to try and growth regulate them. So we believe that it was that mild moisture stress or those drying down events in the substrate that resulted in smaller plants in the peat and coir containers. We don't believe that the peat in the coir containers absorbed any PGR or affected the efficacy of the PGR. Rather, we believe the peat in the coir containers just influenced the plant growth of the Angelonia and Petunia, and simply the smaller plants just required less PGR. So if you're growing in biocontainers that are not very porous, you don't need to worry about adjusting your PGR concentrations. Alternatively, if you are growing in biocontainers that are fairly porous, that are drying down more frequently, you are going to want to look at reducing your PGR concentration in order to avoid over-regulating your crops. So if you take those concentrations of PGR applications that you've been using on plants grown in plastic containers and use them on plants grown in peat or coir containers, you may end up getting super optimal regulation and growing some plants that may be too small or not sized up enough. So you want to look at reducing your PGR concentration when growing plants in those porous containers. There's some acknowledgement that I'd like to give here, and the first one goes to Nick Flax. Nick Flax was a master's student with me for a couple of years. He graduated last year and is now working for the University of Illinois Extension, and this was part of his master project. And so Nick Flax really gets the credit for all the hard work that I get to share with you here today. I would also like to thank the USDA Specialty Crop Research Initiative and NEFA for funding that supported this project. I'd also like to thank Pete Lawler, our greenhouse manager, Kenny McCabe, Kelly Walters, Alex Litvin, Brianna Kenninger, and Erica Schlichty, who are graduate and undergraduate students in the lab who also assisted Nick and I in conducting this research. I'd like to say thanks again to Find Americas for bringing this helping sponsor these webinars so I could bring this information to you. And if Brian will let me, I think I've got approximately 1 minute and 59 seconds for some questions.